Hi guys, so we are back in session 2 of our lesson, the correct use of the microscope. So in the last lesson, that is in the last session, we spoke about what is magnification, we spoke about what is resolution and the factors affecting magnification, we spoke about hand lens, and also I had introduced you to what a compound light microscope is. So today in this video, we'll be talking further about the parts of the compound microscope and their uses and how you could use the compound microscope. And then finally, we'll be talking about the electron microscope. So let's get into our lesson. All right, so as you can see this image, it has been extracted directly from your textbook. So let's read it. Part of the microscope eyepiece and a description, the closest lens to the eye. It is a convex lens. Magnification is shown as times 5, times 10 or times 15. The specimen is magnified by using them. So here is a zoomed in picture of the eyepiece from the microscope. All right, you can take a look at it. So moving on, objective lens. The closest lens to the specimen. They are made of convex lenses. It magnifies the specimen. Mostly, there are three objective lenses that can be identified. They are low power objective lens, the lens with the least magnification, times 4, times 5 or times 8. Mid power objective lens, the lens with the medium magnification, that is times 10, and high power objective lens, the lens with the highest magnification, that is times 40. So here there is a picture of the three different types of objective lenses. So if you look closer, you can actually see the magnification of each lens is stated here in these lenses. All right, so now there are a few specimens in which you can observe it perfectly by using the low power objective lens. So for those specimens, you don't have to use the mid power objective lens or the high power objective lens. All right, but there are some other specimens, all right, if they are really tiny, then you need a better magnification to observe them. So in that case, you can use the high power objective lens or the mid power objective lens. All right, so that is why they have given three different types of objective lenses. In few microscopes, there are even 4 different types of objective lenses. As you can see, this one, it seems to have 4 different objective lenses. All right, so as your will, you can use whatever the objective lens you want to observe your specimen. All right, so it is also made of convex lenses. If you can remember what a convex lens is, it is a curved lens. All right, so now let's move on to the next one. The next one is coarse adjustment knob and fine adjustment knob. So the coarse adjustment knob is used to adjust the body tube to get a clear image of the specimen, and the fine adjustment, it adjusts the body tube finally to get a more clear image of the specimen. All right, so when you use a microscope, we will learn about how to use the microscope in the future. But when you use a microscope, the first adjustment you usually operate is the coarse adjustment. All right, so you can use the coarse adjustment to adjust the body tube, to move the objective lenses lower to the specimen, and for many other purposes. All right, so if the coarse adjustment is not efficient enough, then you can move to the fine adjustment. All right, so by adjusting these two knobs, all right, you can move your stage, you can move your body tube, you can move your objective lenses lower, or even you can move it upwards. All right, so you can adjust the microscope the way you want in order to get a clear image of the specimen. All right, so here is another picture. This is the coarse adjustment and this is the fine adjustment. Okay. Okay, the next thing we have is the stage to keep the slide of the specimen. All right, so here the first image here is a real stage, a picture of a real stage extracted from a real microscope. All right, so the second one is a diagram, but it is more clearer as everything is marked here. As you can see, this is the stage opening. Here's the specimen clip. What is a clip? It is used to keep the slide of the specimen on the stage. All right. So now, usually when you observe something through a microscope, we will learn how to make a slide in the future. But for the moment, there is something called a glass slide. Okay. So on that glass slide, you place your specimen and then you place that glass slide on the stage. Okay. After that, by using the objective lenses, you can observe the specimen on the glass slide. So in order to keep this glass slide steadily on the stage, or else it will move, right? So for that purpose, we use the specimen clip. So you can clip the glass slide on the stage. All right. In that way, you can easily use the objective lenses and observe the specimen. Okay. So this is the stage where you place your glass slide, and here we have the clip where you clip the glass slide. Okay, and here are a few other additional parts in the stage. If you want, you can take a look at that as well. All right, so let's move on. The next one is diaphragm. It controls the amount of light obtained by the specimen. All right, so here is a picture of three different types of diaphragms. Okay, so as you can see, um, a diaphragm is used to control the amount of light obtained by the specimen. A diaphragm is just below the stage. You can clearly see it because it's below the stage. All right. So if you have a microscope at home or even at your school laboratories, then you can observe a diaphragm. But just know that it is below the stage. All right. So it controls the light which reaches the specimen. Okay. So let's move on. So the next one is the mirror. It contains concave and flat surfaces and they focus light onto the diaphragm. Okay, so just for you to know, this is how a concave lens looks like. We already learned about convex lens, so you know that. So here the second image is what a concave lens looks like, all right? So here is a picture of a mirror which has been extracted from the compound light microscope. So the mirror basically, it focuses light onto the diaphragm. We already learned what a diaphragm is, right? So when this light is focused onto the diaphragm is the part which controls the amount of light obtained by the specimen. All right, so it'll be more clear when we learn how to use the microscope. All right, so let's move on. The next one is the base and the arm. All right, so the base, it keeps the microscope on the supporting surface steadily. All right, so that is obvious. The bottom part that supports the whole microscope. And the arm, here, this is the arm, it has been marked. It is used to support the microscope when it is carried. All right, so let's move on. Okay, now we're going to look into the history of the microscope a little bit. All right, so the compound light microscope was first made by Zacharias Janssen. The magnification power of it was 9 times. All right, so here is a picture of the scientist Zacharias Janssen, and this is a picture of the compound light microscope he had invented. Okay, the honor of making a microscope first goes to the Dutch scientist Anton van Leeuwenhoek. The reason is that a world not seen by the naked eye was revealed by this instrument. All right. So though Zacharias Janssen was the first one to invent a microscope, the microscope invented by Anton van Leeuwenhoek was more powerful. All right. So that is why we consider him as the father of microscopy. Many of us don't know that, but Anton van Leeuwenhoek is actually the father of microscopy. So this is a rough diagram of the microscope invented by him, and this is the scientist Anton van Leeuwenhoek. All right. Okay, so now we are going to move on into the correct method to use a compound light microscope. So this image again has been extracted from your textbook. So let's read one by one, all right? The first one, first keep the microscope on a horizontal table steadily in a place where strong sunlight will not fall. Okay, so as you can see in this image, the microscope is placed on a horizontal table steadily, all right? use the cos adjustment knob to bring the low power objective lens down. So I already told you that the cos adjustment and fine adjustment knobs can be used to adjust the microscope as you will, alright. So the cos adjustment, mainly we use it to move the objective lenses. So the first thing we are going to do when we use a compound light microscope to observe a specimen is that we bring the low power objective lens down. As I already mentioned, first, we use the low power objective lens. Alright, if you think the image is still not clear, then we move on to the mid power objective lens and finally the high power objective lens. Alright, so the first thing is to do is to use the cos adjustment knob to bring the low power objective lens down close to the specimen so that you can get a clear image. Alright, so as you can see, this is the cos adjustment knob. This person is rotating it in order to move the low power objective lens downwards, all right, close to the specimen. Okay, adjust the mirror and the diaphragm to obtain a clear spot of light by keeping the eye open on the eyepiece. All right, so the mirror and diaphragm, they work together. I already said that, right? So the mirror focuses the light on the diaphragm and the diaphragm controls the amount of light which is to be absorbed by the specimen. All right, so we can adjust the mirror and diaphragm until we get a clear image, but while doing that, we need to keep our eyes on the eyepiece. All right, only then we can see, right, what is happening to the specimen. Is it clear or is it still not clear? Okay, so we should make sure that we are observing the specimen through the eyepiece while adjusting the mirror and the diaphragm. All right. Fix the prepared slide on the stage using clips. All right, so that is pretty simple. You can see that we have made the specimen here, and now by using the stage clips, we have fixed the specimen on the stage. All right, I will teach you how to prepare a microscopic slide in the next slides. All right, in the upcoming slides. For the moment, just remember that you make the slide and then you fix it on the stage. Okay. The fifth one, bring the low power objective lens upward using the rough adjustment until a clear image is obtained. Make sure that both the eyes are open during the process. Keep the comfortable eye at a distance of 1 centimeter to the eyepiece. Alright. So rough adjustment is the cos adjustment, if you are wondering. Alright, it's just another name. So we need to bring the low power objective lens upward using the rough adjustment until a clear image is obtained. All right. I already mentioned that we can first we move the low power objective lens downwards in order to get a clear image of the specimen. And now you can again move it upward if you want to, make some alterations in the image you are seeing. All right. Make sure that both the eyes are open during the process. Yeah, if your eyes are open, only then you can see the image, right? And you need to keep the comfortable eye at a distance of 1 centimeter to the eyepiece. All right. So you need to keep in mind that you shouldn't like fix your eyes flat on the eyepiece when you are observing a specimen. All right. It is not the correct method. You need to keep your eye in a considerate distance to the eyepiece. All right. Only then you can get a clear image. Okay. The next one, try to obtain a clear image using fine adjustment. A sharp and clear image could be obtained by adjusting the middle and high power objective lens if necessary. Yeah, as I mentioned, if you cannot get a clear image in the low power objective lens, you can use the middle and high power objective lenses if necessary. So the low power objective lens, it actually gives you the widest view. All right. By that, it allows you to slowly bring the object into focus. So that is why we basically first use the low power objective lens. All right. If that is not enough, you can move to the middle and high power lenses. Use the rough adjustment to bring the low power objective lens upward after the observation. Okay. Why do we bring the low power objective lens upward again? This is a very important point. The reason why you move all your objective lenses, not especially the low power objective lens, but any objective lens that you have been using to observe the specimen, first of all, you need to move it upwards. The reason is, when you are going to remove your specimen from the stage, if your objective lens are down or really close to the specimen, it might contact the objective lens. All right, that is, the objective lens and the specimen might touch each other if the objective lens is close to the stage. That is why we use the rough adjustment to first move the objective lens upward, and then you have to remove your specimen from the stage, and you use a dry piece of cotton cloth to wipe the stage. All right. So that is the correct method to use a compound light microscope. Okay, so now let's look into the factors to be considered in the use of a microscope. Alright, the first one, when the microscope is carried from one place to another, the arm of the microscope should be held by your familiar hand and the other hand should be kept below the base of the microscope. Then hold the microscope towards your body. Okay, basically, it's like this. So as you can see, image B is the wrong way to hold the microscope and image A is the correct way. All right, you can see now the arm of the microscope should be held by your familiar hand. Yes, this person is holding the arm of the microscope with his familiar hand, and the other hand should be kept below the base. The other hand is below the base. Then hold the microscope towards your body. Yeah, you can see he's holding the microscope towards his body. Okay, so let's move on to the second one. Wipe the stage using a clean piece of cotton after using the microscope. Okay, so you don't necessarily have to use only a clean piece of cotton while you're wiping your microscope. You can use a clean piece of cloth or even a clean piece of tissue, all right? And the most important thing you need to remember is that before you clean the microscope, you need to make sure that your hands are clean. All right, because you might have touched the table, you might have touched other lab equipments, so your hands might be dirty, right? So first of all, you have to wash your hands well, and only then you have to start cleaning your microscope. Okay, so this one is a air compressor. All right, so you can buy air compressors from the markets. So you can use an air compressor to remove the dust particles from a microscope. And also in the pharmacies or even the markets, you can get lens cleansers. So you can use a lens cleanser to wipe the microscope and also the objective lenses. Okay, so let's move on. So the third one, if the microscope is not used for a long period of time, the lenses should be removed and placed inside a desiccator with silica gel or anhydrous calcium chloride. Okay, so what is silica gel? Silica gel is a desiccant or drying agent that manufacturers often place in little packets to keep moisture from damaging certain food and commercial products. Okay, almost every one of us, we have seen silica gel before. Okay, do you remember seeing tiny non-edible packets placed inside biscuit packets or chips packets? So those packets contain silica gel. So we put this silica gel into these products in order to absorb the moisture in the area. So if you are wondering what is moisture, moisture is the water diffused in small quantity as vapor. Okay, so if there are water particles in a food product or other commercial products, it can damage the product in many ways. So to prevent that, we add silica gel packets to absorb those water particles or moisture. Okay, so now I think it is clear for you. So what is the use of silica gel in the storage of microscopic lenses? In regions where humidity is high, if you are wondering what humidity is, humidity is a concentration of water vapor present in the air. Okay, so in regions where humidity is high, fungus development is common with microscopes lenses, which causes lens deterioration. Deterioration means damage. In order to prevent this, desiccants such as silica gel can be used to absorb the moisture in the container. Alright, so when humidity is high in a particular area, fungus development arises. So this fungus development can cause harm to the microscopic lenses. So we add silica gel, which is a desiccant or a drying agent which will in turn absorb all the moisture in the container. Alright, so let's move on. This is a picture of dry silica gel desiccant. Alright, so you might have seen this in many commercial products. And the second picture here is anhydrous calcium chloride. So anhydrous calcium chloride can be found in the laboratories. This again is a desiccant just like silica gel. Okay. So the fourth one, the microscope should be stored in an upward position to minimize the accumulation of dust. So that is simple and clear. All right. So you can also use like a polythene bag in order to, store the microscope in a safe place, and then you have to keep it in an upward position. All right. Fifth one, use clear slide and cover slips in the observation. So here is the part where I explain to you how to make a microscopic slide. Alright, so first of all, what is a slide? A microscopic slide is a thin flat piece of glass used to hold objects for examination under a microscope. Alright, so here is the picture of a microscopic slide. Alright, so whatever your specimen is or any object that you're about to observe under a microscope, you need to place it on this glass slide. Okay, and then you fix this glass slide on the stage of the microscope for observation. Moving on, what is a cover slip then? A small thin piece of glass used to cover and protect a specimen on a microscopic slide. Okay, this is how a cover slip looks like. Okay, so now basically when you place a specimen or an object on the glass slide, sometimes dust particles may fall into it, or even microorganisms may contaminate the specimen. So to prevent that, we use something like a covering. That is the cover slip. All right, it is a thin piece of glass used to cover and protect a specimen on the microscopic slide. So you use a cover slip and you place it flat on the specimen so that the specimen gets protected from dust particles or even microorganisms. Okay, so basically it's like this. This is a glass slide. As you can see, you place your specimen on the glass slide. This is the specimen, and this is the cover slip. So you use the cover slip and you cover up the specimen, and then you place the glass slide in the stage for observation. Okay, so now I think it's clear. Sixth one, do not change the lenses of one microscope to another. So when you change the objective lenses of one microscope to another, sometimes there might be errors in your observation. So to prevent that, it's best not to change the lenses from one microscope to another. So, calculation of magnification power of a microscope. Magnification power means the number of times that the specimen is subjected is to be magnified. Okay, so already I explained to you what magnification is, right? So it is the process of enlarging an object. Okay, so or the process of enlarging the original size of an object is called as magnification. So you can calculate the magnification power by this equation. You multiply the magnification of the eyepiece by the magnification of the objective lens. Simple as that. So if you are asked to calculate the magnification of a microscope, you simply multiply the magnification of the eyepiece and the magnification of the objective lens. Let's look into an example. The magnification of eyepiece was 20 times and the magnification of the objective lens was 30 times in a particular microscopic observation. What is the magnification of the microscope? So simply you multiply the magnification of the eyepiece by the magnification of the objective lens. So 20 times 30 is 600. So the magnification of this particular microscope is 600. Alright, so let's move on. Let's look into another example. The magnification of eyepiece was 10 times and the magnification of the objective lens was 40 times in a particular microscopic observation. What is the magnification of the microscope? So you simply multiply the magnification of the eyepiece by the magnification of the objective lens. That is 10 multiplied by 40, you get the answer as 400. Okay. When you are observing a specimen under a microscope, sometimes we draw linear diagrams, also known as line diagrams. At such instances, we have to mention the magnification of the linear diagram as follows. Magnification of the eyepiece multiplied by magnification of the objective lens again multiplied by number of times the visual image is magnified when drawn. So basically, when you observe anything, let it be a specimen or any other material, you need to roughly draw what you see through the microscope. So this rough drawing is called the linear diagram. So when you draw a linear diagram, you have to make sure that you mention the magnification of the linear diagram. So this is how you do that. This equation is how you mention the magnification of the linear diagram. Okay. So this image has been extracted directly from your textbook. As you can see, the Rio lower epidermal tissue and the low power of microscope. So Rio is actually a plant. So this is the linear diagram of this image. Okay, as you can see, it looks pretty much the same, but this is a rough drawing. Okay, so when you draw a rough drawing of the specimen, you need to mention its magnification as such. This equation, magnification of the eyepiece is 10, magnification of the objective lens is 4, and number of times the visual image is magnified when drawn is 3. So as you can see, the magnification of this linear diagram is clearly stated. Okay, so let's look into the second figure. It is the stained human cheek cells under high power of light microscope. So already in here they have given us, right, the magnification of the eyepiece and the magnification of the objective lens. So you just need to multiply this part by 2. So what is this 2? It is the number of times the visual image is magnified when drawn. So this entirely, that is 10 multiplied by 40 multiplied by 2, is the magnification of this linear diagram. So to understand this further, let's look into certain examples. The magnification of the eyepiece in a certain microscopic observation is times 20. The magnification of the objective lens is times 5. And the number of times the visual image is magnified when drawn is 2. Write down the magnification of the linear diagram of the above specimen. So we simply follow this equation in order to obtain the magnification of the linear diagram. What is this equation? Magnification of the eyepiece multiplied by magnification of the objective lens multiplied by number of times the visual image is magnified when drawn. Okay, so now you just extract the data from this question. Magnification of the eyepiece according to this question is 20, so you write 20. Magnification of the objective lens according to this question is 5, so you write 5. And finally, the number of times the visual image is magnified when drawn is 2, so you write 2. So 20 times 5 times 2 is the magnification of this linear diagram. So a similar example, the magnification of the eyepiece in a certain microscopic observation is times 15. The magnification of the objective lens is times 4, and the number of times the visual image is magnified when drawn is 3. Write down the magnification of the linear diagram of the above specimen. So simply you multiply the magnification of the eyepiece by the magnification of the objective lens, and finally by the number of times the visual image is magnified when drawn. That is 15 multiplied by 4 multiplied by 3. So this is the magnification of this particular linear diagram. Okay. So finally, we are going to talk about the electron microscope. Okay. The primary difference between the electron microscope and light microscope is that an electron beam is used in the electron microscope whilst light rays are being used in light microscope. We already learned that in compound light microscopes, we use light for observation. But in an electron microscope, we use electron beams. Alright. The maximum magnification power is 500,000 times. The maximum resolution power is about 0.0005 microns. This is about 200 times higher than the healthy human eye. Virus is the smallest microorganism in the planet which cannot be observed under a compound light microscope. Only an electron microscope can be used to observe a virus. So when you read this description, you might get the idea about how powerful the electron microscope is. It is even more powerful than the compound light microscope because the virus, which is the smallest microorganism, you cannot observe this under a compound light microscope, but you can observe it under an electron microscope. So you can analyze how powerful the electron microscope is. Okay. Here we have two pictures of an electron microscope. You can pause the video and take a good look at it. As you can see, it looks like it has a lot of parts when compared to the compound light microscope, right? So just take a look at it. So now let's look into the instances where an electron microscope can be used. First one, to observe the activity of pathogenic microorganisms such as virus and bacteria during research. What is pathogenic or pathogenicity? Causing a disease or the ability to cause a disease can be defined as pathogenic or pathogenicity. Alright, so to observe the activity of disease-causing microorganisms such as virus and bacteria during research, we can use an electron microscope. So this is a picture of AIDS virus under the electron microscope. Here we have bacteria under the electron microscope. Okay, just for you to know. To learn about the internal structures of cell in detail. What is a cell? Cells are the basic building blocks of all living things. So you will learn further about what is a cell in your upcoming lessons and your upcoming grades. But for the moment, you just have to know that cells are the basic building blocks of all living things. Okay. So cells are really tiny. You cannot see them with your naked eye. But even inside a cell, there are some internal structures. So the first image here you can see is a plant cell. And this second image is an animal cell. Okay. You will learn about these in your upcoming grades. But anyway, as you can see, even inside a cell, there are many internal structures, right? So you can use an electron microscope to study these internal structures inside the cell. Okay. The third one, to use in genetic research. You will learn how that is done in your upcoming grades, okay? And the fourth one, to get information on crime investigation. I can give you one example, that is fingerprints. Some suspects in a crime scene, they leave their fingerprints, all right? So experts can use a microscope, an electron microscope to observe these fingerprints, okay? So that is one example I can give to you. So finally we've reached the end of our lesson, guys. I hope this video was informative. So comment down below if you have any questions or any suggestions on how I could improve my lessons, and like, subscribe and share the video with your friends, guys. And thank you for watching.